Okay, wonderful. So hi everyone, and welcome to our webinar today on gut health and arthritis research updates. So my name is Jess and I work with our health promotion teams in Arthritis Queensland and Arthritis New South Wales. And I'm really excited to have three guest speakers with us today talking about the links between our gut health and arthritis, as well as our overall health. So they'll share with us some of the research that they have been involved in, and we'll hear about how we can influence the health of our gut. And we've got a really nice mix of some science and maybe some sort of complex terms that will hopefully be explained to us all, as well as some practical information of what we can do. So just a quick disclaimer. That, as always, the information presented in our webinars series is general information and advice and does not substitute for the individual advice of your health team. So always consult with your healthcare provider for individual medical advice and treatment. In the spirit of reconciliation, Arthritis Queensland and New South Wales acknowledge the traditional custodians of country throughout Australia and their connections to land, sea and community. We pay our respect to their elders past and present and extend that respect to all Aboriginal and Torres Strait Islander peoples today. So I'm very excited to introduce our first guest speaker today, Dr. Anne-Sophie Bergot, an expert in immune regulation and microbiome at the University of Queensland Translational Research Institute in Brisbane. So after receiving her PhD in immunology and physiopathology from La Sorbonne Universitas in France, Dr. Anne-Sophie debuted in Australia in 2011 to better understand immune regulation in HPV-associated cancer and in type 1 diabetes. In 2007, she was awarded the Jian Cho Research Excellence Award, and she then continued her journey working on immune arthritis, mentored by Professor Ranjani Thomas, supported by Arthritis Australia. Her interests focus on deciphering the crosstalk between the gut microbiome and the innate and adaptive immune system and how these interactions lead to the development of autoimmune arthritis. So I'll just hand over the controls to Dr. Anne-Sophie and she can take it away.
Good afternoon, everybody. Let me just check if I can see. Oh, I can't see the pointer anymore, actually. Here you go. All right, so I'm very pleased to present you with the work we are currently doing in Ranjani Thomas group at the University of Queensland, Tierra Institute in Brisbane. And I'll start my talk with a general explanation of the topic. And then in the second part of my talk, I will be more in detail into the science, and I'll try to explain as much as I can the sometimes complicated projects. So first of all, what are the inflammatory big family of arthritis disease? Inflammatory arthritis affect the joint and the related structures, and it involves different conditions. You have rheumatoid arthritis or RA, systemic lupus erythematosus or SLE, gout sometime, and spondyloarthritis SPA, which is the disease that I'm studying here in the lab. Today, there is no cure. The current treatment aims to relieve pain and stiffness, prevent and delay disease progression and additional complications, even spinal deformity. The treatment is lifelong, and the biologic disease-modifying drugs do not suppress all symptoms. Patients have to stop therapy due to adverse events or loss of efficacy. So there is urgent need for new therapy options, of course, and for this very important healthcare and socio-economic issue. Where is it now? Oh, here we go. So there are some factors associated with this complex arthritis disease. And the big ones are the main known ones are genetic factors, and the big gene complexes associated with the disease are HLA gene. The HLA genes is the master histocompatibility complex, or MHC, that was first identified and remains the most studied region. It accounts for 1/3 of the disease genetic susceptibility. In RA, it is known that HLA-DRB1 carrying patients have the most risk to develop RA, and in SPA or ankylosing spondylitis, it's HLA-B27. Apart from these HLA genes, you also have non-HLA genes that are mostly related to immunological pathways. And among the most represented ones, you have defective antigen presentation, inflammatory cytokines that are mediators driving inflammation, and microbiome control. And we'll touch a bit of all of that during my talk. So besides genetic factors, you also have lifestyle risk factors, and you have quite a few different ones. One of them is the smoking, and the smoking is linked to the lung organ in the body, right? Then you have the diet that is linked to usually westernized diet or not very healthy diet. And here the mucosa that are involved is the oral mucosa as well as the gut. You also have airborne agents that can increase the risk of developing arthritis. And here again, the lung mucosa is the one that is in contact with these agents. You also have infectious agents such as viruses and other infection, bacteria or parasites. And here, pretty much all mucosa are infected: the gut, the lung, the skin, the vagina, oral mucosa. And overall, the microbiota as well will be shown in this talk and by Mark that it can have a lot of good or bad roles in the development of autoimmune disease, and it's linked with oral and gut mucosa as well. So mucosa and bacteria is one of the highlights of this lifestyle risk factor. So just to go a bit deeper in the oral hygiene and rheumatoid arthritis, it's been shown for a long time that periodontal disease, or PD, is a common disease that is in pretty much half of the population, and it results in breaches of the oral mucosa, leading to gingival bleeding with translocation of oral bacteria to the blood. It's more common in patients with RA, so it's been said to be the cause potentially of RA, and RA patients have ongoing PD with increased disease activity. I just come back from a conference in Italy where I met Dana Orange, who made a massive, tremendous breakthrough in science showing that this disease was actually potentially very likely to be one of the causes of rheumatoid arthritis. And Ranjani as well published similar results a couple of years ago. And the gut barrier integrity is also a massive topic in the field because people with excess weight, obesity, Western or poor diet have increased gut permeability, and we'll show later in my talk that this is a massive correlated problem in the development of autoimmune disease, and often comes with the dysbiosis, gut dysbiosis, which is defined by an imbalance of the populations of bacteria that are in your gut. You can have good bacteria, you can have bad bacteria, but the problem is when you lose the good bacteria and you have more of the pathogenic bacteria, you have this dysbiosis that is associated with gut inflammation, loss of barrier permeability, and then problems. Sorry. But there is hope, of course, obviously, luckily. These pathobiomes that are growing can be suppressed. And if you encourage the healthy commensals to take back their place, you can reverse this dysbiosis and come back to the balance. The bad guys, they thrive on sugar, and sometimes you can find that in the gums in your mouth or in the gut. And they will eat that, and they will eat the mucus that is meant to preserve the barrier integrity. So lower sugar would be better. And the commensals, the good ones, they love fiber, they love fruits and vegetables. And there was a talk again in Italy where this young clinician showed that the vegan diet was actually good. The problem is a lot of people dropped because it's a very hard diet to follow on the long term. But I'm pretty sure Hannah will give you a nice talk after that about the diet in arthritis. I don't know if it's hard to pass. Okay. So gut microbiome, I mean, the microbiome in general is very, very important. And as I just said, the gut microbiome is everywhere. You have a skin microbiome, you have a gut microbiome, you have an oral microbiome, you have a urogenital microbiome, and all of them have different roles, pro and good, and they are meant to be keeping you healthy. But when you have disbalances, they can go to diseases. So what is the microbiome? That's a very brief slide because I'm sure Mark will be a lot deeper in this topic. The microbiome, you have different phyla that are separating the big families of bacteria. Then you go to a class that divides everything. And then again, the order, the family, the genus, and then the species. And some of the species in red have been shown to be associated with bad prognostic in autoimmune arthritis, and the one in green are usually the ones that are the good ones that need to be enriched or present and are often deficient in the people with arthritis. And the one I'm studying myself are in yellow, Parabacteroides goldsteini and Lactobacillus murinus. So overall, here to finish this global introduction, the mucosal barrier dysfunction can be oral or lung or gut, may facilitate the spread of insoluble microbial remnant into the joints and causing disease. Controlling this mucosal inflammation barrier dysfunction is genetically predisposed individual may reduce the dysbiosis and the propensity of arthritic development, and that's what we are studying here. So overall, better oral hygiene, stop smoking, better diet, more exercise, and less stress is a good combination to be in the right favor. So, but what about the immune system and immunotherapy in context? So as I said earlier, I'm studying spondyloarthropathies, which are mainly seronegative arthropathies that include several chronic inflammatory arthritic disease that affect the joint. It's strongly associated with HLA-B27, and as opposite to RA, it's characterized by the absence of rheumatic factor. We have axial spondyloarthropathy or ankylosing spondylitis that affects the spine, and we have peripheral spondyloarthropathy that are affecting small joints and also are called psoriatic arthritis that affect also the skin, reactive arthritis, which is usually caused by the infection bacteria like chlamydia or gonorrhea in the vaginal tract, and some arthritis with or without gut symptoms like IBD. But the common symptoms still between these two axial and peripheral diseases are extra-articular symptoms that affect the gut, the eye, and the skin. So what is the gut-joint axis in SPA? We know that the healthy population, pretty much 40% of the global population are healthy gut. But, 40% of the patients with SPA have a healthy gut, but about 50% of them have subclinical gut inflammation. So sometimes it comes without symptom, but it's there on the microscopic level. And 10% of these patients are also having IBD, which is inflammatory bowel disease, usually Crohn's disease. So the intestinal microbial dysbiosis occurs in SPA, but we don't know today if that's a cause or an effect that drives the disease. So the past research has focused on profiling the immunological and microbial changes in SPA. And studies have been shown that the gut of SPA patient is mostly leaky. We said leaky gut because we know that it's lets something cross from the inside of the gut to the internal part of the gut. It's that the study of the microbiome here is done by 16S sequencing, which gives you access to the species level of the bacteria that I showed you earlier. And we can also detect this leakiness of the blood through the leakiness of the gut through the blood by measuring some compounds that are released from the gut through the blood. We know that in the synovium, in the joint, inflamed joint of patients with SPA, we find T cells that are the main immune cells that are usually cytotoxic. And they are here to normally fight the bad guys, but they end up in the joint where they cause inflammation. And it's been shown last year that if we deplete this T cell, we can achieve remission in patients. So it was a very good hope that we are on the right track for research. And of course, how do we go from the gut to the joint? The hypothesis that we have in the lab is that it goes through the blood. And again here, without going into detail, some markers from the gut have been found in the blood and then found in the joint as well. So there is a good pathway from one to another. So our lab aims to unravel the mechanistic link between these dysbiosis, the gut and the joint. And the hypothesis that we have is that the gut-derived pathobionts are taken up by some cells that are eating these bacteria. We call that phagocytosis. And then they are displaying the bacterial antigen to the T cell. They travel through the blood and they end up in the joint where they reactivate the T cell that causes arthritis. We use a mouse model in the lab called SKG. It's a very good model that mimics human SPA. When it's usually healthy, but when we give them a trigger, it develops all of the symptoms of SPA. It develops arthritis in the joints and the tail. It develops a gut inflammation with swollen of the gut. It develops, by histology, we can see a lot of infiltration of immune cells in the tail, in the rear Achilles tendon, for example. In the gut, we have massive infiltration, and we also see psoriasis-like lesions on the ears here. And they also have sometimes uveitis as well. So it's been extensively studied in this paper here a few years ago by Ranjani Thomas. We know that these mice have a fecal dysbiosis with enriched gram-negative bacteria, and they have also increased production of pro-inflammatory cytokines that are key in the SPA. So as you can see here briefly, that's the epithelium of the mice. This one is the mice that can be sick. This one is the mice that can be resistant to the disease. And here we just see the histology. So we see there is, for the first week, there is no signs of disease. But when we look at the bacteria in the villi of the gut, the bacteria are circled in white. And you can see that day 2 after the trigger, it's the same between the two mice. Day 4 after the trigger, the mouse that is susceptible to disease have a lot more bacteria within the villi. And at day 7, it's again the same, while this one seems to be resistant. So why that? Why are these bacteria crossing the villi and going in the gut? So we used to study this dysbiosis in these SKG mice. And as you can see here, that's representative of dysbiosis. As I said before, we have an enrichment in gram-negative bacteria, which are usually associated with bad guys. And the most abundant one is called Parabacteroides. And that's the one I will study later. When we treat the current treatment to the mice, which is anti-IL-23p19, which is the same drug as that is given in human, you have a massive color here, which is the Clostridiales here, which is associated with good bacteria. So we can reverse this dysbiosis with the treatment, having good bacteria, and then we protect the mice from developing symptoms. So this dysbiosis normal balance are very important. So I'll come back to that mouse, to that bacteria here, and we are studying the role of that only one bacteria in gnotobiotic SKG and biopsy mice, which are germ-free. They have no bacteria at all, so we can play with that and just put one bacteria in and see how it works. So how can gut-derived bacteria trigger SPA? So here at TRI, we have an animal facility that have mice with the full flora, but we also have a gnotobiotic facility. Just to show you how it looks, we have germ-free mice in those isolators here, and we put the arms in here, the black gloves, to enter the bubble and being able to work with the germ-free bacteria. That's we have a few different ones here, and when usually these isolators are for germ-free mice, and when we recolonize the mice with bacteria, we transfer them to racks where they are still isolated for the rest, but still not associated with the full flora. So what do we do with these mice? So we give them Parabacteroides goldsteini or Lactobacillus murinus, or they remain germ-free, and then we can compare these 3 different mice. We gavage them orally, and then the bacteria colonize the gut. And later we have a look at, we give them the trigger, and we look later at the scoring, the histology, and how are the bacteria behaving. So we know that this one here is very pathogenic. It drives severe ileitis and arthritis. Here you can see the villi are really nice and normal, while here we have a lot of purple infiltration within the villi. And in the peau, it's the same. It gives a lot of purple things here that are infiltration of the immune cells. And it's also very swollen compared to here. So we can visually measure that as well in the mice. The ileitis is measured by weight loss. So we weigh the mice and we know that they lose weight because they are not able to function enough. The Lactobacillus murinus induce milder ileitis and arthritis, but we also show that it can promote gut resilience. And when it's combined with Parabacteroides, it can actually mitigate the effect of Parabacteroides. So we have these 2 good and bad bacteria that we can play with. And when the mice stay germ-free, they develop no ileitis and no arthritis. So this proves that gut bacteria, because that's the only bacteria in the mouse, are driving the disease. So that's a very important part. And so we use this model to analyze the immune pathways, myeloid cells, T cell. So Parabacteroides goldsteini acts as a pathobiont, and Lactobacillus murinus acts as a probiont in the absence of the trigger, the cordlan, and it's been shown this one to mitigate the symptoms of Parabacteroides. So how is this seen by the immune system? What are the T cells doing, and what are the antigen-presenting cells doing? So here we again use the germ-free mouse, gavage with Parabacteroides. We give the trigger, and then we look at different lymph nodes along the tract from the gut to the joint. So we start with the gut, the mesenteric lymph node drains the gut, and then the spleen is the filter of the body. So everything goes through the spleen usually. And then we go down to the popliteal lymph node, which drains the foot. And then we look at the foot, the tail, and the gut itself. And we do that by imaging using a microscope. So first on T cell, what do we see? You can see this bar here in black are all higher than the small ones here. The small ones here are the proliferative T cell in the biopsy, the resistant mouse, and all these ones here are activated and proliferating T cell in the SKG susceptible mouse. And you can see it's true for all spleen, mesenteric lymph node, popliteal lymph node, and lumboaortic lymph node that drains the tail. It's true for this population of Treg cells, which are normally protective, and it's true for the non-Treg cells that are normally cytotoxic, CD8 T cells that are cytotoxic. So this means that T cells are proliferating in the SKG mouse that has Parabacteroides, but not in the biopsy mouse that has Parabacteroides, which means in the context of a disease-prone mouse, the T cells are hyperreactive and proliferating in response to the bacteria. And we can also observe that in the ankle. So the conclusion was, can Parabacteroides be a source of antigen for those T cells? And is that by mimicry, because the T cell in the SKG mouse are autoreactive. They are not meant per se to react against your bacteria. They are meant here to protect you against viruses or attacks, but there is some dysfunction in the T cell that makes them react too much against something that they're not meant to. And we don't really know why still. So then I said that the bacteria was translocating to the villi, and we can see here again in yellow here, you see the signal of the bacteria within the villi of the mice. And they actually co-localize with macrophage that are in purple here. So you see the purple macrophage in the villi of the gut, and it co-localizes with the signal of the bacteria in the gut. It's absent from the biopsy mice, or I mean, it's not absent from biopsy mice, sorry. So it's there. It's much reduced when the mouse are treated with the drug that protects them, reduced here, but it's still happening in the biopsy mice, although to a reduced number. And this showed us that the macrophages that are meant to eat up these bacteria in the gut, they are here. But this one here in the SKG mice are associated with inflamed gut, while this one here in the biopsy-resistant mouse are associated with normal gut. So they have different function. Some are associated with inflammation and some are associated with normal. So they could have two different phenotypes. In the ankle, we also see this colocalization of macrophage with the bacteria, which means that we can track the macrophage carrying the gut bacteria in the ankle. So that's very important. And then, because we think it goes through the blood, we had a look at the spleen, which is the filter of the blood. And you can see with the yellow dots here, that's all the bacteria present in the spleen. And they are in the area that is pink here, which is usually full of macrophage. So here again, we think that the macrophage carry the bacterial signal from the gut to the spleen to the joint. So when we further look at this macrophage, I said to you that you have potentially two different phenotypes, some good guys, some bad guys for the cell, the macrophage. So we had a look at that in the mouse again, using a technique that is called flow cytometry that allows us to have multiple parameters on the cell surface. And we analyze the cells one by one and check what do they express. And then we can compare between organs and between mice. So this gave us figures like this that shows different populations of macrophage depending on the markers that they express. And I highlighted two here because they are very interesting for us. That's this one here where the red is increased. The red is the SKG mouse. And here the blue is increased, which is the biopsy. So when we look more in detail, we can see that the population 9 here is here, and the cell displays these markers, and these markers have been associated with good macrophage, the one that are in the resistant mouse and the one that are meant to remove the bacteria from your system and let you in peace. The population 2 here is, however, increased in the SKG mouse, but not the biopsy mouse, and express markers that are associated with inflammation and have been reported in the literature as the bad guys, the one that drive inflammation and the one that should be potentially driving the autoreactivity. So our susceptible mouse has less of the good guys and more of the bad guys. So what does it do when we inject these ones in the mouse again? So here we sorted these cells, we sorted the macrophage, we sorted them. So the cell sorter is like we use the marker, the cell surface, and we can isolate from the tissue the cells in a tube, and then we can use this for different analysis. And because they are live cells, we can re-inject that to mice and see what they do to the mice. So we used different donors for that. We used the disease mouse, SKG SPF, SKG, the resistant mouse biopsy, and the germ-free SKG mice. So basically what we want to do is we want to inject these cells to the mice. They are injected to SKG mice because they are the mice that are prone to be developing the disease. We've labeled the cells, so you can see the site of injection here. And we injected close to the paw because we wanted them to be going to the popliteal lymph node and then activate T cell there and then drive arthritis. And this mouse here was our control without cells. And what we've seen is that the macrophage that are the bad ones coming from the SKG mouse donors, when they are injected to SKG mouse recipient, they can drive disease. That's visual arthritis. The paw is inflamed. While the macrophage that are coming from the resistant mice biopsy injected to SKG recipient here in blue, they have delayed or pretty much significantly lower arthritis, and the ankle stays normal visually. And the macrophage that come from germ-free SKG mouse, they don't develop arthritis. So this tells you that macrophage coming from SKG disease mice can drive the arthritis, and they need to have seen the bacteria to be able to drive arthritis. So it gives you the link that the gut bacteria in the gut, they are taken up by these macrophages. They travel through the blood, via the spleen, to the lymph node where they activate the autoreactive T cell. And then they are able to, in the ankle, drive arthritis by reactivating those T cells. So the conclusion overall, take-home message, is that in at-risk patients that express HLA-B27 in SPA, they have a gut inflammation, which is often coming with dysbiosis, enriching gram-negative bacteria. These bacteria can cross the villi because the gut is permeable. And in here, they come into an environment that has a lot of inflammatory macrophage and less protective macrophage. And also, I haven't touched about that, but deficient T cell. So the bacteria are taken up by these macrophages. They travel through the blood, and they end up in the spleen, in the bone marrow, and the lymph node, where they can activate T cell. So the M1-like macrophage, they carry the microbial antigen, display that at the cell surface, present this to the cell for gut bacteria-specific T cell, and these T cells that also come from the gut end up in the bone for some reason, where they are activated and drive inflammatory response and then bone destruction. And with this, I want to thank Ranjani Thomas for the opportunity to work in her lab, and our student, Ben Kite, who did a lot of imaging, Mark Morrison for his support and help in this project, and everybody else, Jack Biggin, which is an expert in IBD, inflammatory bowel disease here at the Mater Hospital, and all the facilities at the TRI Fraser Institute for the imaging, the flow cytometry, and the microscopy, as well as the animal facility, and of course, Arthritis Queensland and Arthritis Australia for their support in this research. I hope it was okay. I'll take your question after the other talks. Thank you.
Yeah, thank you, Anne-Sophie. And yeah, if anyone has any questions, just pop them in the chat and we'll get to those after. But thank you so much, Anne-Sophie. That was such a great introduction to the gut microbiome and a really great peek inside, first of all, what's happening inside our bodies that we never get to see, as well as what you and many other researchers are doing on a daily basis to better understand that link. I'll just... Okay, so our next guest speaker for today is Professor Mark Morrison, who leads the microbiology and metagenomics group. In 2013, Professor Morrison was appointed chair in microbial biology and metagenomics at the University of Queensland Diamantina Institute, which is based at the Translational Research Institute. Here, he and his team are using DNA sequencing technologies as well as other microbiological and genetic methods to produce new insights into the microbial world. Oh, I've just sort of gone a little bit too far. And I'll just hand over control to Mark. Okay, Mark, you should have control of the keys now. Just unmute.
I've got a... Can you hear me now? Is it okay?
Yes, okay, we can hear you.
Okay. Yeah, well, good afternoon, everyone, and it's a pleasure to follow on from Anne-Sophie. And I've just got my disclosure slide here. I think I have PhD training in microbiology with no medical qualifications. Okay, so... I'm having this up. Okay, so just a little bit of background information. Hopefully I'll go over very quickly. We do live in a microbial world, and the human body serves as a scaffold for the collection of distinct populations of microorganisms that colonize different parts of our body. And the major groups of these bacteria, at least, are listed here. And there's really only 6 major groups, and I might use an iceberg as a bit of an analogy. So The 6 names you see at the bottom of this slide would be the tip of the iceberg, but underneath each of those tips is a vast amount of undescribed information, which can be really quite large in size. Most of the iceberg we don't see from above the surface. And I want to talk a little bit about that and its relevance to health and disease. Many of you may think the skin actually is the largest surface area of the body, and it's about the size of a tablecloth, but it's the gut environment that really is the residence of a very dense population of microbes. In excess of 100 billion microbes per teaspoon of gut contents in some parts, especially the large intestine that Anne-Sophie is focused on today. And the gut itself, if you were to look at its surface area, would be about the size of a tennis court. Okay, so it's really a much larger surface area where host response and biology and the immune system can interact with this very dense and diverse population of microbes. And so there's many more microbial cells than there are human cells, and we may be considered to be like a superorganism, a combination of both. Now, what's been the challenge? Like I said, those 6 major groups might represent the tip of the iceberg, but for microbiologists like myself, over a course of 20 or 30 years, it was what was underneath the surface that was hard for us to define and characterize, because we really couldn't grow many of these organisms in culture in the laboratory setting. So how did we get around that process is by the opportunity revealed by something called metagenomics, and I'll give a brief definition of what that is in a moment. Metagenomics is to look at the entire community without first having to culture them. So a couple of the ramifications that are important for patient health in a clinical setting is that we can start to collaborate with clinicians and the support of stakeholders and consumers in various areas of the health sector, and work with real-world samples for the first time. And as a result, using these culture-independent ways in DNA sequencing, we confirm and validate not just the complexity and the diversity of these microbial communities, but for the first time give us a bit of a blueprint in terms of what they're actually capable of doing. So I was asked to give you some concepts around the composition and the function of the gut microbiome, and I'll try and do that with the rest of the time I've got. So how do we do this? Persons like Professor Thomas or some of my gastroenterology colleagues will provide us with stool samples or a buccal swab from the cheek, or biopsy that might be collected during endoscopic procedures. And rather than trying to initially grow the microbes in the lab, we extract all the DNA from that particular sample. And then if you look in the direction of the upward facing arrow from that sample, we can produce through various techniques something of a stamp collection of the types and kinds of microorganisms that are there. And what that facilitates is shown in the top panel. We can use those stamp collections and their similarities and differences, and by statistical techniques, show whether there is a clustering between groups of individuals according to the parameters or clinical measures that are made for those persons. So imagine that this may be in green, a group of persons suffering from rheumatoid arthritis, and on the right-hand side, it may be a first-degree relative that doesn't have rheumatoid arthritis. And we're looking at the stamp collections of microbes in their stool samples, and we see a clear separation between the two. When you look at the downward leading arrow, we use very high throughput recent DNA sequencing technologies. And rather than just looking at a single specific piece of DNA, now we build an entire picture of the genetic potential of that complex community. And we've done that for all of the microorganisms because we haven't biased our sample with culture. And what we produce is this kind of what I call a heat map in the bottom right-hand panel. And each row would represent a particular gene, and each column would represent a particular individual. And you can see that the top left is red, and the bottom right is red, and the top right is blue, and the bottom left is blue. And so you can picture in your own mind that persons with rheumatoid arthritis are shown in the top half of the figure, and they have genes with functions of high content in that metagenome in red and a low content in first-degree relatives that don't have rheumatoid arthritis that is blue, and vice versa. So we can start to see that the functional attributes, in addition to the types and kinds of microbes, differ between these individuals and whether they suffer from something like rheumatoid arthritis or not. And this is kind of the holy grail, to identify whether the microbes, as Anne-Sophie has started to elegantly show, can play a role in triggering rheumatoid arthritis or arthritises of different types in response to the genetic susceptibility of those individuals. So Anne-Sophie used the word dysbiosis, and I just want to give you a so what and who cares about dysbiosis very quickly. And so the microbiota profiling that's been done in persons suffering from a recurrent form of gut inflammation, or they're a person that's just been newly diagnosed as having inflammatory bowel disease or rheumatoid arthritis, if we look at their samples of stool, we can see two key changes in the population that are inherent to this term dysbiosis. First of all, we see an increase in the abundance of bacteria that are listed here in red. And Anne-Sophie's made reference to some of these organisms, Bacteroides or Parabacteroides, for instance, as having a deleterious effect. Many of the people perhaps listening are perhaps familiar with these names because these bacteria have been recognized for a long time as being potentially infectious, pathogenic bacteria that can drive inflammation and infectious disease. But the other key feature to point out here is that coincident with the increase of those shown in red is a decrease in the abundance, both in a relative term and absolute term, of the bacteria that are shown here in green. And I'm pretty sure most of you don't recognize these particular names. And importantly, what these organisms represent are the ones that have been very difficult to culture historically, and we've been able to identify them in some level of clarity through DNA sequencing approaches. And these bacteria are often decreased in persons suffering from inflammation that might be associated with rheumatoid arthritis. And they're mainly found in high relative abundance in persons without symptoms or a phenotype of that particular type. So why is that important? Well, it's when a person has a high abundance of those bacteria shown in green that the left-hand panel is most relevant. In other words, we don't have a leaky gut. We have a thick mucin layer that serves as an additional protective barrier. And there are microorganisms that are supported by the diet that are numerically abundant and members of those groups that I showed you in the previous slide in green. But what happens with inflammation and dysbiosis is a depletion of those good guys, and as Anne-Sophie pointed out, an increase in the abundance of the bad ones. And the net result is as the middle panel shows, an inflammatory response, a thinning of that protective mucus layer, ultimately with overt inflammation being gone altogether, and the gut being leaky, and the translocation of these so-called bad bacteria coincident with that situation. So I guess the point I'm trying to make here is that when we talk about dysbiosis and we talk about some of these chronic conditions, of which rheumatoid arthritis is one, it's not just about a germ theory of infection and inflammation. It's something that's more holistic and changes in the overall community population makeup and the functionality associated with those organisms. And it's not just an increase of the bad guys, but a depletion of the good guys. And so the battle, so to speak, is to understand how we can tip the balance back towards the ones in green and thereby deplete the ones in red and promote a greater resilience or a reduction in symptoms, or perhaps even a reversal and cure of some of these chronic inflammatory conditions. Okay, I won't dwell on this model, but I came to Diamantina, as it was called, in 2013 to collaborate with Ranjini in particular and Matt Brown in their studies of arthritis and various forms of arthritis, and contributed to some of the work that showed two things which I think are important to share with the audience today. I've lost, there we go. The first is that, as Anne-Sophie pointed out, the mouse model for arthritis emphasized here in Brisbane with the Thomas group is the SKG mouse model. And when that mice, those mice are kept germ-free or naive of microbiology, they don't develop the overt inflammation. So they have the genetic susceptibility, but they don't appear to have the microbial stimuli to drive the inflammation forward. And it's only when those SKG mice have bacteria colonizing their gut that we start to see these inflammatory responses. So this is a demonstration early on that's supported, or is the foundation of Anne-Sophie's work, is to try and identify who are the key players that might be driving that inflammation in the microbial world or who isn't. And the validity to support that is shown in the panel on the right. So in the SKG mice that are kept separate from the control mouse group, when we treat them with the curdlan as a trigger of inflammation and they have microbes in their belly, we see a high level of inflammation in the SKG mice, but none in the control group of mice. But when you mix those two groups together and you treat the arthritis model, mouse model, with the SKG to drive the inflammation, you can see that the lines or the individual mice really develop no inflammation at all when they're co-housed with the other control mice. So what does this mean? What it means is that the control mice have members of in their microbiota, in their bellies, that through the behavior of the mice when they're together in the cages rather than separate, are sharing microbes between each other. And so the control mice have some protective microorganisms that can be transferred to the arthritis model and blunt the inflammatory effect. So this is a foundation of Anne-Sophie's work, not just to look for a bad guy like Parabacteroides, but start to identify some of the good bacteria that we can find in healthy mouse models and hopefully in the human subjects that help to blunt these sorts of interactions with the immune system in persons suffering from these conditions. So really, metagenomics and the approaches that I've tried to summarize very quickly emphasize the commensal microbiota, bugs that have historically been recognized as commonly being there, but not really shown or known to have key benefits. But this summary of this picture shows that commensal bacteria provide a lot of protective, metabolic, and structural functions that are important in promoting health. And that we really need to think more about the only good bug being a dead bug and a germ theory of chronic inflammatory diseases like RA, to thinking more about the population of microbes as a community and promoting the good guys and getting rid of the bad ones. Okay, so very quickly, I just want to acknowledge two groups of people. Obviously, thank you to Arthritis Queensland and New South Wales for the opportunity to speak with you, and importantly, your consumer and stakeholders that enable the capacity for my clinician colleagues shown here on the left to do the work that they do, and the opportunity for basic scientists like me and my colleagues shown on the right to try and characterize the microbiome like Anne-Sophie is doing, and identify not just the missing microbes, but various ways we can use this information to design new types of consortia for what people might call fecal microbiota transplant, which might be a question or two later, to develop dietary strategies, perhaps, that promote the growth of good bugs and downplay the growth of the bad ones, and also to help us identify strategies whereby we can work with people to help manage their diet in an effective way with the expertise of Hannah to move ourselves forward and build resilience. So I just want to finish by saying, who would have thought that we'd think about stool samples from laboratory waste trucks as being a gold mine of new biomedical insights and innovations? We still really lack the capacity to see what's around the corner. But I think the point I'm trying to make is with the support of Arthritis New South Wales and Queensland and your consumer and stakeholders, it's a great opportunity for someone like me to try and identify new ways to try and help and thwart the issues and difficulties people that suffering from rheumatoid arthritis have. And just to finish my part of the section, with Anzac Day tomorrow, I just acknowledge all those that have served in our Defence Forces, lest we forget. Thank you.
Wonderful. Thank you, Mark. Such a great introduction to learning more, not just about the bad bacteria, but also the good bacteria, and like you explained, that there's the community that we have to be thinking about. So just on to our last speaker today, Dr. Hannah Mayr is an advanced accredited practicing dietitian and works as the Principal Research Fellow for the Nutrition and Dietetics Department at Princess Alexandra Hospital in Brisbane. Dr. Mayr has clinical research expertise in chronic disease prevention and management, and her work has a focus on evidence-based dietary patterns, particularly a Mediterranean-style diet and anti-inflammatory approaches. She's part of a team of medical and allied health researchers investigating novel ways to manage rheumatoid arthritis through lifestyle intervention. Dr. Mayr is an experienced university lecturer and regularly educates doctors and nurses on diet's role in chronic disease management. Thank you, Hannah.
Thank you so much for the introduction, Jess, and the opportunity to be involved today. Jess, I'll just check with you. What would you like me to do from a timing point of view, since we're edging towards 1?
Yes. If you have capacity to just stay on for an extra 5 minutes or so to go through your presentation, that would be wonderful. If anybody does need to jump off at 1, that's completely fine. But I think a lot of people will be eager to stick around and hear from you and see your whole presentation if suitable.
Yeah, great. No, just wanted to check, and more than happy to go through the slide content and stick around. That's fine. So I'll change tack a little bit, and I'm going to be talking a little bit less about some of the science and a little bit more about where does this lead us in terms of dietary intake, which both Anne-Sophie and Mark have linked in the concept of how diet is ultimately a huge impact on our gut, and that has an influence on arthritis. I'll just check. Oh. There we go. So I'll talk very briefly about sort of where dietary evidence is at from the perspectives of inflammation, the gut, and arthritis. Then I'm going to focus in on some really practical, hopefully, takeaways for you in terms of, well, what are the actual key eating principles that we know of that can help promote health in these areas, and then some tools and resources that may be useful for you to help you to think about those eating principles. So on the slide here, summarising where we're sort of at in terms of evidence for overall dietary patterns and whether they're related with inflammation in the body. So when I talk about a dietary pattern, what I mean is the amount, variety, and combination of different foods and drinks that someone has. And so on the slide here in red, we have what's called sort of a Western dietary pattern. And this is what is known to be pro-inflammatory. So this includes things like processed foods, refined carbohydrates, high salt, high sugar, high meat. Then in the yellow color, we have diets that are focused on individual nutrients like low fat or low carbohydrates. And diets that just focus on these nutrients really are sort of inconclusive or have neutral effects on inflammation in the body. But in green, we see the types of eating patterns that have evidence to support that they are anti-inflammatory. So these are things like a Mediterranean style diet, the DASH diet, or vegetarian. And I'll go into the principles of these later, but what we do know about all these patterns is that they're highly plant-based. So where are we at in terms of evidence for dietary interventions in arthritis? So at the moment, we mostly have small intervention studies in both osteoarthritis and rheumatoid arthritis with some promising results around disease activity and symptoms. However, we do have a lack of large controlled studies in these disease areas. But this doesn't mean that we can't make diet an important part of prevention and management. We know diet impacts on inflammation and it can improve quality of life, overall health, heart health, which is an important factor to manage in people with arthritis. And also there are good studies to show diet can improve mental health as well. So where we sort of sit in terms of current reviews or guidelines, where we know is that ideally you want to focus on a dietary pattern that can be aligned to sort of an anti-inflammatory eating approach. So those Mediterranean plant-based, or you might hear the term prudent eating patterns. And like I said, I will also go through those principles later. But what I've also got on the slide here is that it's actually really important to consider individualization and whether someone is able to sustain the eating principles that they're aiming to improve in their diet. So in terms of gut health, something that's really important to consider is plant food diversity. So we've heard from our other speakers about the diversity that we have in the microbes in the gut. And essentially we know from some preliminary evidence that, to properly feed the gut, you need an abundant and diverse range of fiber, which comes from plants in the diet. So the more diverse diet you have of different plant fibers, you tend to have a stronger gut microbiome. And a really interesting study, which I've cited on the slide here, was in more than 1,500 people where they characterized usual diet as well as had fecal samples to look at the gut microbes. And what they showed was that people who had 30 or more different types of plants in their diet per week versus people who had only 10 or less different types of plant foods in their diet per week had a more diverse gut bacteria. And they also showed that they had less antibiotic resistant genes. So this leads us to going, we really need to increase how many different types of plant foods are in the diet. Another study that's been recently conducted in 5 European countries is called the New Age study. And in over 600 people, they looked at a year-long intervention using a Mediterranean style diet approach. And this is one of the first studies out there that really has got robust data on how a dietary intervention over time might influence the gut. So what they actually showed was adherence to the dietary intervention led to an increased abundance in specific types of microbiota in the gut. And these different microbiota that were changed were associated with markers of lower frailty and some improvements in cognitive function. And they were also associated with inflammatory markers as well. So less inflammatory markers if you had this good change in bacteria. And what this really supports us to know now is that it shows there's some feasibility in changing someone's usual diet to a more healthy eating pattern, which influences the gut microbiota and then has the potential to promote aspects of healthier aging. Now, this is a complex slide where I don't expect you to understand everything that's on it, but there's definitely some similarities here to some of one of the slides that Mark showed. So hopefully you can understand a little bit about what you're looking at in terms of this representing the gut and the lining of the gut. And on the left-hand side, it's essentially trying to summarize part of what we know around following a healthy diet aligned to that sort of Mediterranean style diet approach that has good fibers, micronutrients, high quality fats, And you see inside the cells there, it's saying that they're maintaining healthy microbiota and good barrier integrity of the cells. And then that leads to better immune tolerance, so better immunity and lower inflammation. Whereas on the right-hand side, we have that term Western diet again, which is more like your, diets high in red and processed meats, lower quality fats, refined carbs, salt, etc. You can see the lining of the gut is different. There's more gaps, and there's that concept of leaky gut or increased permeability. And then there's that term, this leading to more gut dysbiosis, like Mark referred to. And what that ends up leading to is more inflammation. So what I've summarized very briefly for you is a little bit of a taste of where are we at in terms of inflammation, gut, and arthritis, where there's key linkages between those three things. And really where the evidence leads us is some key eating principles that might help to assist, prevent, or manage having, good gut health and also considering arthritis. So a key tip is eating plenty of vegetables, fruits, and whole grains. So these have a suite of different nutrients and components to them, like antioxidants, that are healthy for multiple body functions and overall health. Ideally, you want to aim for diversity and variety, both across the fruits and veg and the whole grains, and try to include them across the day in your diet. Choose healthy protein sources. So if we're thinking more plant-based, this means including foods like legumes more often. So aiming for 3 or more times a week, including fish or seafood, especially oily types like salmon, tuna, mackerel, sardines, because they have omega-3. Having nuts most days, which are a really great source of fiber as well as different bioactive plant compounds, but also nuts are a really good source of unsaturated healthy fats. Limiting red meat intake and limiting processed meats. Use herbs and spices to flavor foods. So we know that herbs and spices can have anti-inflammatory effects, and also including them increases the flavor of foods and also helps to reduce salt intake. So on the slide, I've just listed some examples of herbs and spices that are known to have anti-inflammatory influences. Include dairy foods, mostly fermented. So unfortunately, dairy foods have a history of being a bit attacked, and people tend to unnecessarily exclude them from their diet, but they actually can have anti-inflammatory activity. And especially fermented forms like yogurt and cheese can be beneficial for your gut microbiome. And you really only need to choose reduced fat varieties if you are managing your weight or your cholesterol. Use extra virgin olive oil. So I've referred to here about extra virgin having a higher content of polyphenols, and polyphenols are essentially bioactive compounds found only in plant foods, and the more refined an oil is, the more it loses those polyphenols. So extra virgin is the least refined oil. And importantly, a lot of people think that you should be choosing things like light olive oils because they have less calories, but really it actually is a more refined and lower flavor oil, not about the calorie level. Olive oil margarines only have about 20% olive oil in them, so you're actually much better off using an actual extra virgin olive oil rather than using spreads. And importantly, you can use it in cooking. That's a real common myth that we have to bust. You can use it both raw and in cooking. The household temperatures we use for cooking don't tend to reach levels that cause the oil to oxidize, like some people commonly believe. And you can actually look, in Australia, there's the option to certify your oil and have a label like I've showed on the slide. So you can actually look to varieties, look if your olive oil that you have has that label on it or not. Portions on a plate. So I'm sure many of you have seen these sorts of concepts before, but they're a really great practical tool to think, okay, when I'm eating a meal, how am I actually combining different foods, including plant-based foods, in a way that promotes more intake of vegetables, being half of your plate, versus about a quarter of the plate being your whole grains, a quarter being your protein sources, and then thinking about, are you flavoring it with things like extra virgin olive oil and herbs and spices? So I would challenge you to, when you have your next meal, maybe you're going to go eat lunch after this session, what is your ratio of protein foods to veggies to grains, and think about trying to increase the portions like this plate. Another great concept you can think of is the plant to animal ratio in the foods you eat in your diet. So in a typical Australian diet, a lot of people have a ratio of around 2:1. But what we ideally want to be aiming for, if we do choose to include some animal foods, is a ratio of more like 4:1. And you can see an example meal on the slide here, where you've got a little bit of feta cheese and a little bit of red meat making up about 20%, but the rest of it comes from 80% of plant food. So that's another way of thinking about, across your diet or your meals, how much of it is coming from plants versus animals. And obviously plant foods can still be unhealthy, so it's about those healthy sources of plant foods. Now, a really great concept that has been coined, I guess, by Dr. Megan Rossi, who is commonly referred to as the gut health doctor, who's actually a dietitian by background and a researcher. But you would have recalled that I referred to a study that showed if people ate 30 or more plant sources of foods a week, they had better gut diversity. And so taking from that research, this is basically a way of setting yourself a bit of a goal to say, well, could I have 30 or more different type of plant foods in my diet on a weekly basis? And 30 sounds like a lot when you first say that, but hopefully this slide might give you a bit of inspiration of where you can get diversity from, because every different type of vegetable or different type of fruit, whole grain, legume, or nut and seed counts as a point. So this highlights that not only should you be thinking about having, say, fruits, vegetables, etc. in your diet, it's also about different types of fruits and vegetables, because they all offer different nutrients and different types of fiber, etc. So another challenge you could really think about is, write down all the different types of plant types of foods you eat in a week and think, does it actually hit the 30 or is it under that? And where could I potentially diversify some of the foods I'm having to hit those targets? And also thinking about, which of these plant food groups are you missing? Some people don't eat many legumes or don't have nuts. So are they the ones that you could target more? So just to round out, I wanted to, direct you to some existing freely available resources that would provide you with some practical tips that align to the evidence and the eating principles I've just been through. So I work in Queensland Health, and we have a suite of free resources directed to different, conditions or disease areas. We are referred to as the Nutrition Education Materials Online, or NEMO, but there is a specific Mediterranean style diet resource on there that I actually helped develop that. So literally, this is just some snips of what the resource looks like. But if you actually were to just Google NEMO Mediterranean Diet, this would be a resource that would come up as a PDF you could download. The National Heart Foundation has some really excellent up-to-date information around this type of healthy eating pattern. So they have some website content as well as downloadable PDFs. I've shown on the slide here that they have this easy URL you can use where you just put hrt.how/eating, and that will take you to their healthy eating information. They also have a really great suite of healthy recipes. So essentially all their recipes have been created with the same key healthy eating principles in mind, like I have shared with you today. They also have e-booklets of their recipes that are themed. So for example, like on the slide, they have an e-book with about 5 or 6 recipes in it based on 5 ingredient recipes that are quite simple. Or they have budget recipes or a range of different things. So I really would encourage you to check out their resources. The Baker Heart and Diabetes Institute also have a range of up-to-date fact sheets. So if you were to Google Baker fact sheets, it would come up with their pool of fact sheets. You just need to filter to their nutrition specific ones. So I would suggest they've got ones around making healthy meals, plating it up. So the similar plating portions I directed you to, just with some more practical components in there. And then also things like healthy snacks. So you can even see on this slide that the sorts of things that are being promoted across those resources align with those different healthy foods that we talked about. It's just more practical tips of how to actually include it in your diet. In terms of books or recipes, I've just got some suggestions on the slide here. Catherine Itsiopoulos was my PhD supervisor. She's Greek by background and has led a lot of Mediterranean diet research in Australia. The first two books were created by her, and the third one is by some dietitians who are based in Brisbane. And then the fourth one I've got on there is actually, there is a Grain and Legumes Nutrition Council in Australia. And again, if you just Google GLNC or GLNC recipes, it'll take you to their website. And really, this is really useful for yourselves, or if you know people who are trying to increase intake of legumes. I know a lot of people aren't really sure how to include legumes in their diet, so that can be really useful as well. I've also just put on there Old Ways. It's an American-based website, but they've got a whole lot of really good recipes and information around these types of eating patterns as well. And I've referred to earlier Dr. Megan Rossi, who has used the title The Gut Health Doctor. She is Australian. She actually did her PhD with PA Hospital, where I now work, but she is based in the UK now, and she's very well known and has some great books, and also has resources on her website, including a gut health quiz as well. So really useful resources there. And the final slide on tips for me would be seek support from a dietitian. So we don't expect you to be able to navigate all of this on your own. Dietitians are trained to help people to individualize these approaches. And so ask your GP about having a dietitian as part of your management plan. If you do have osteoarthritis or rheumatoid arthritis, for example. And Dietitians Australia do actually have a Find a Dietitian search tool on their website. And you are able to filter to the people on there that specifically indicate they specialize in arthritis or, for example, gastrointestinal disorders as well. So thank you so much for the opportunity to share, and I look forward to any questions, depending on what we have time for, Jess.
Yeah. Wonderful. Thank you, Hannah. Some really great resources that you've shared there that I'm excited to go and check out myself. Just stop sharing. Sorry. Okay. I think I've stopped sharing. So unfortunately, I apologize. We are a little bit over time, and some of our guest speakers may need to jump offline. We may have capacity to maybe do a couple of questions if anybody and any of our speakers are able to stick around, but also understand if not. I do have one question to start off, and I'm not sure who it may be. It might be Mark, but really interested to know, is it are there methods for people with arthritis that are reliable and I guess easily accessible for people to find out more about their individual gut bacteria? And is it worth finding out, or should we just be sort of putting some of those practices into place to improve our overall gut health?
Yeah, thanks, Jessica. Look, it's a little bit of both. There are a number of companies now, some based in Brisbane, some in New South Wales and Victoria, that offer a service to produce an assessment of the microbiota in your stool sample in particular. Many of the services that are currently available give you something of a stamp collection. The one in Brisbane does the more detailed analysis and gives you information about the wiring diagram, the functional characteristics. Mm-hmm. I think at the moment, while these data sets are useful to compare you to other people. Their application and use in a clinical setting is still premature. They're of value in terms of helping you identify if you change your diet, for instance, to one like Hannah has suggested, what sort of changes you may see in your stool microbiome. These aren't cheap tests, and they're not PBS listed, as I understand, but that's how they would be used. But importantly, I think if persons feel comfortable with providing that sample and collecting that information, it does help these sequencing services and their basic scientists learn a lot more about what are the biomarkers that might be present in stool that can be used in a diagnostic way, or even what might be called prognostic, used in a predictive way to say, hey, you're starting to see some changes here. It might be time for you to think about your food choices and to change your dietary pattern and try and move back towards a microbiome that favours good guys rather than an outgrowth of bad ones. Okay, so nothing of any strong medical utility at the moment, like a diagnostic test for infectious bacteria, but the knowledge that is being created by supporting those services can provide a future where they have more clinical relevance. Yep.
Yep. Wonderful. Thank you. And I think you mentioned you may need to head off. So if you want to jump off, that's completely fine. Thank you so much again, Mark, for joining us today and for your presentation. Really helpful.
Yeah. Look, it's been a pleasure to be involved, and I look forward to future opportunities, engagement with Arthritis Queensland and New South Wales as people may find beneficial or useful. Yep. Thank you. Yep. Thank you, everyone, for your attention today.
Thank you. Hannah, there was a couple of questions that I think would be relevant to you. What someone commented about trying to include a lot more vegetables and fruits and grains in their diet, but also struggling with maintaining weight. Yeah, do you have any sort of tips or suggestions around that?
Yeah, and I think, weight's something I didn't touch on because it is relevant at both ends of the spectrum, but I guess we want to try sometimes take the focus off weight as well. But definitely if someone's trying to maintain their weight, what we would sort of look at is the concept of high energy or kilojoules and high protein sources, but healthy high energy and protein sources. So we would still always promote the inclusion of foods like vegetables, which do tend to have less kilojoules or calories in them, but it's about replacing other aspects in the diet that can help maintain the kilojoule content. So things like extra virgin olive oil, for example, or nuts are really healthy, but actually relatively high in kilojoules. So they actually can be a really important inclusion in the diet alongside things like vegetables that could help someone to maintain their weight. And I guess it does need to be individualized and very much will depend on what someone's usual intake is. The other aspect of it is about regularity of eating as well. And so things like how often in the day are you having meals? And some people, if a goal for them is to need to maintain weight, it's about having smaller frequent meals, for example, rather than 3 larger meals across the day. And yeah, so density of the food and also how much you're having. So definitely can still work within those healthy food sources, just including both the lower and higher calorie components as well.
Yep, wonderful. And probably one more question is probably all we've got time for. But we often hear a lot and see a lot of ads around prebiotics and probiotics and how they can help our gut health. But I noticed it wasn't something that was mentioned. Are prebiotics and probiotics useful? And yeah, sort of where do they play a role?
Yeah, so Anne-Sophie, feel free to jump in here too. But I guess from a practical point of view and just to explain to the audience in terms of pro versus prebiotics. So probiotics are the actual live bacteria and they're present in certain foods like your fermented dairy. So, certain forms of yogurt and things like cheese have live bacteria in them, as well as some of your other fermented foods like sauerkraut and kimchi. But then you've also got prebiotics, which are essentially components that feed your bacteria.
Yep.
And so prebiotics are actually found in a really large range of foods, and essentially most plant foods that have fiber have some levels of prebiotics in them. So definitely consuming prebiotics is part of the recommendations aligned with what I talked about. Probiotics and whether or not to take a supplement versus, foods, etc., is a huge area and hard to make strong conclusions for, because in the literature, it's hard to decipher between what's been studied, because there's a lot of different strains of bacteria and different types being studied in different supplement studies, and then also whether or not those different strains have been studied for particular outcomes is really varied. So my understanding is that we don't have, strong evidence yet to say if you consume XYZ bacteria in this supplement, you're going to have this effect on your arthritis. We don't have that level of evidence yet. But I guess what we do know is that it's unlikely to be harm related to taking probiotics. So if people wish to do so and can afford to do that, there are potentially beneficial effects. How much, we're still not quite sure. I do know that the gut health doctor, or Megan Rossi, has some blog articles on her website about probiotics use. And she actually has, she does say on her website that she's going to be holding a webinar later this year where people can sign up their interest in that topic. So if people are really interested in probiotics, they can sign up through her website to do that webinar later in the year.
Yeah, wonderful.
So if I can add on that, so in the lab we've been comparing a non-pathobiont Parabacteroides and a probiont Lactobacillus, and Lactobacillus is meant to be the one you can find, for example, in yogurts. I mean, it's good bacteria, but the thing is, if you put it alone in the mouth, it will still drive arthritis and ileitis because it's alone there. So it's here in high number and there's nothing else to balance this one. So the key is the balance. If you mix the two together, then you can start seeing the beneficial effect of this normally probiont. So I would think having probionts, some probiotics sometimes on a short period of time can be okay. But you can imagine if you eat too many of them every day for years, you will go towards disbalance because you will push these ones over the other ones. So even though you think you are increasing the good ones, you are in disbalance. And this disbalance is harmful. So the key is to be in balance. So having a bit of them is good, not too much, not too often. And having, I think prebiotics is better, because you feed them all. You just add one, or the probiotics, you don't, it's still a restricted amount of different kind of bacteria. So prebiotic will feed a lot more diverse probionts and therefore keep the balance better. So
Wonderful. I think that's a really good way to sort of end and summarize all the presentations today, is really focusing on that balance. And Sophie, when you sort of showed all the different strains of the bacteria, and Mark talking about the balance of the good and bad, and Hannah about the variety and balance of different diet components, perfect way to summarize today. So we will have to finish up. We have gone way over time. Thank you so much, Hannah and Anne-Sophie, for joining us today, and Mark, who has jumped offline. Such a really interesting topic, and great breadth of knowledge, sort of looking in that scientific and what the researchers are currently working on, as well as a great range of practical information, Hannah, and more resources that we can all go and look into. So thank you again so much.
Thank you.
Thank you.
Thanks everyone for joining us today. Enjoy the rest of your afternoon.
Thank you.